Welcome to Biochemistry Unfolded, from atoms to algorithms, an online podcast on the topic of biochemistry and biophysics. Let's think about this question. If an AI predicts a perfect biomolecular structure, but it misses the real-world physics by up to 30 degrees, is it actually a discovery, or is it just a really good guess? Welcome back to Biochemistry Unfolded, from atoms to algorithms. And today I'm pulling back the curtain on AI-driven molecular design. And we all know tools like AlphaFold have revolutionized biology, but a fascinating new study proves that when it comes to nucleic acids, these algorithms are often just playing an imitation game. They're copying what they've seen rather than understanding the chemistry of life. So today let's look at how researchers have used solid-state NMR to expose where AI stumbles and why flexible RNA loops are the ultimate algorithmic nightmare, and what this means for the future of medicine and biotechnology. And the paper I'm reading this week is a 2025 study by Christina Bergonzo and Alexander Grishaev entitled Critical Assessment of RNA and DNA Structure Predictions via Artificial Intelligence, the Imitation Game. And this was published in the Journal of Chemical Information and Modeling on March 30th, 2025. The full citation is in the video description below. Now, while artificial intelligence tools such as AlphaFold have shown massive success in predicting protein architectures, and they have the potential to model all of life's biomolecules, their accuracy when predicting nucleic acids such as RNA and DNA remains highly variable. While you might think that it can't be too hard to predict the structure of a simple double helical molecule with just 4 base pairs, the world of nucleic acids is actually far more complicated. DNA can also form triple helical structures with Hoogsteen hydrogen bonds, as well as 4-stranded structures called G quadruplexes and I motifs. And especially these are common in telomeric DNA. Single-stranded RNA is conformationally flexible, and it can fold in much the same way as a protein. And this constitutes the structure of the ribosome, transfer RNA, even ribozymes that can also catalyze reactions. So there's certainly a lot of interest in obtaining structural data on DNA and RNA. And of course, from an evolutionary perspective, we're also interested in exploring the origins of genetic information. Was DNA or RNA the original genetic molecule? So in this study, the authors emphasize that AI oligonucleotide predictions often lead toward mimicking structures found within their training data sets rather than accurately reproducing underlying physical realities. Hence the phrase in the title, the imitation game. AI tools generate models that can accurately predict simpler architectures, such as canonically paired helical stems, but they severely struggle with structurally complex and dynamic elements. These problem areas include internal loops, capping loops, non-canonical base pairings, as well as areas possessing high conformational flexibility, all of which are vital for RNA folding, biological, and cellular functions. And many of these same issues in prediction are similarly applied to protein structural prediction as well. Furthermore, one of the key problems with AI structure prediction is that for many simple proteins, it is a very good prediction, at least when comparing the backbone structure. Side chains are a completely different story entirely. Yet, because the predicted structure is so good, it's easy for researchers to fall into a trap and think that it must be correct, even for more complicated parts of the biological macromolecule. Yet we can't forget that the AI models used are often based by the training set it was provided. It's comparing structural information from known experimental structure databases, and it's arriving at conclusions based primarily on that data. The missing component that many AI models neglect is the consideration of the biological and chemical data that contributes to the structure. Things like ion concentration, pH, molecular crowding effects, and a whole host of other things. Another major issue is that biological macromolecules often have more than one conformation, especially molecules that are highly flexible. Yet AI will focus on the conformations available based in the training set. It tends to be biased by what's there. So it may miss the mark entirely here. Now, the authors of this paper utilize the new AlphaFold 3 model, which is available on the Google DeepMind AlphaFold's server, and that's alphafoldserver.com. To understand AlphaFold 3, let's first look at a little bit about how AlphaFold 2 works. And I've got tons of other videos on how AlphaFold 2 works. But let's think of AF2 as a master linguist and an architect specifically for proteins. You give it a 1D string of amino acids, and that's the primary structure. And then by looking up evolutionary history and how amino acids talk to each other, it predicts the 3D coordinates of the protein's backbone. And it does that with incredible degree of accuracy. Now, AlphaFold 3 transitions from a protein architect to an all-in-one molecular matchmaker. It doesn't just fold proteins. It models how proteins interact with DNA, RNA, chemical drugs such as ligands, modifications like phosphorylation. It can handle certain cofactors and ions like magnesium, zinc, etc., all simultaneously. And it treats the entire cell matrix as a deeply interconnected puzzle. But these differences aren't just minor software updates. Google DeepMind completely swapped out the entire underlying engine of the AI. First, by going from a residue-centric to an atom-centric model. AlphaFold 2 looked at the world in residues. It simplified its math by treating every amino acid as a rigid single block, a token, and then calculated the angles of the peptide, the backbone phi and psi angles, and the side chains. Those are the chi angles. And so this works beautifully for proteins, but it fails completely if you try to input a zinc finger domain bound to a zinc ion or a small molecule drug. The AI wouldn't know what angles a free-floating ion has. AlphaFold 3 also throws out the residue-only rulebook, and it tokenizes standard amino acids and nucleic acids normally. But it can break chemically modified components or small drug molecules down atom by atom. And so this atomic atom-centric flexibility follows AlphaFold 3 to model literally any combination of biomolecules found in the protein data bank. Secondly, the structure model was replaced with a diffusion model, the module, right? And so AlphaFold 2 uses a highly specialized structure model that mathematically forces the peptide bonds to stay joined in a rigid chain while rotating the bonds to find the final shape. AlphaFold 3 replaces this entirely with a diffusion model, the exact same kind of generative AI used in image creators such as DALL-E, or protein design tools such as RF diffusion from the Baker Lab. And so it starts with a cloud of completely randomized chaotic atomic dust, pure noise, and it gradually pulls the atoms into focus over multiple iterative steps, and it's guided by the chemical sequences that you gave it until you get a crisp, high quality definition 3 molecular interaction. And lastly, AlphaFold 3 emphasizes multi-sequence alignments, or MSAs. AlphaFold 2 relied heavily on the MSA, but it had to scan massive databases of evolutionarily related sequences to figure out which residues co-evolved. And that means that if residue A mutated into something positive, residue B mutated into something negative to maintain an ionic bond. Processing these giant MSAs took a massive amount of computational power. AlphaFold 3 uses a streamlined module called the pair former. It de-emphasizes the heavy reliance on massive MSAs, making it much faster and much more data efficient, while still picking up on the cross-molecular relationships between entirely different types of chains, such as a transcription factor binding to a specific promoter sequence on the DNA. As revolutionary as AlphaFold 3 is, you always have to keep the physical ground truth in mind, because AlphaFold 3 uses a generative diffusion model. It can occasionally suffer from what AI researchers call hallucinations. For example, if you give it a highly dynamic, intrinsically disordered protein region, or a highly flexible RNA loop, like the ones Bergonzo and Grishaev evaluated, AlphaFold 3 might confidently draw a neat static structure where in reality the molecule behaves like a floppy shifting noodle, all in solution. It's an extraordinary tool for generating hypotheses, but solution-state biochemistry experiments like NMR or cryo-EM are still the ultimate judges of truth here. So let's look at figure 1 in the paper. And so this illustrates a specific famous piece of RNA, and this is called a stem loop 5, SLV, and this is from the ribosome. It's not just a standard double helix, but it contains a highly specialized structural motif known as a U-turn. That's a UNR motif. And in biochemistry, a U-turn is a tight loop where the RNA backbone sharply bends backwards. Because of this tight turn, the structure is incredibly sensitive to its chemical environment, specifically the cations, the positively charged ions that are dissolved in the surrounding water buffer. Figure 2 uses a side-by-side structure comparison to show exactly how the RNA changes shape based on whether magnesium or sodium ions are present, and then compares the experimental reality of NMR with the AlphaFold 3 AI predictions. In a real solution containing only sodium and no magnesium, the RNA loop is relaxed and loose. There's not enough concentrated positive charge to counteract the negative charges of the phosphodiester backbone. So the loop spreads out. It lacks a crucial hydrogen bond between the 5' phosphate of the base of the uracil and the uridine residue. And then when the authors asked the AI to predict the structure using only sodium conditions, the AI completely missed the environmental context. Instead of generating the loose loop, it predicted a tight, compact U-turn. It basically guessed what a generalized ideal U-turn looks like from its training memory and then ignored the specific low salt biochemistry of the solution. And when a divalent cation like magnesium or manganese was added to the real tube, the +2 charge was bound to specific pockets, and that neutralized the negative charges, and it tightly locked the RNA into a canonical, highly compact U-turn shape. So you would think the AI would do better here, since it wants to draw a compact turn. However, when the authors added magnesium ions into the AI simulation, the algorithm choked on the coordinate details. It placed the magnesium ions in entirely incorrect positions, and they didn't match any of the real coordination chemistry sites found via NMR. In some variations, adding the ions actually caused the AI to lose the neat base tacking directly following the turn. Now, if we cross-reference this figure with table 1 in the paper that has structural parameters such as the turn residue alpha angle and the base stacking distances, we can quantify how much the AI was off by. While the real NMR structures show a massive shift in the backbone alpha angle when you swap out the ion environment, shifting from a loose about 116 degree to a sharp approximately 184 degree angle, the AI predicted shapes that stayed rigidly stuck, hovering around a fairly tight turn regardless of what ions were floating around it. This is actually proof of environmental ignorance. The AI knows what an RNA double helix looks like, and it knows what the average loop looks like. But it doesn't understand the thermodynamic relationship between an RNA backbone and local buffer conditions. It plays the imitation game, and it draws a pretty picture of what you think you want to see rather than calculating the true biochemical physics of the solution. So now let's look at some of the terminal sequences. As is often the case with proteins, the ends of DNA and RNA molecules are highly dynamic, while the middle of a double helix is locked down tight by cooperative hydrogen bonding and base stacking. The terminal base pairs undergo a process called fraying, kind of like a rope, if you will, right? Where the ends kind of fray across, and get frazzled. And so they naturally breathe, unzip, and zip back up again in solution. Figure 2 actually illustrates the apparent systemic failure of the AI to model the local sequence dynamics and structural fraying at the terminal ends of the nucleic acids by comparing computational models against experimental NMR data. While solution state biochemistry shows that terminal base pairs are highly dynamic, flexible, and responsive to sequence mutations, the AI models consistently overstabilize these regions, and that predicts them as idealized, rigidly stacked helices. This occurs because the AI's training data relies heavily on X-ray crystallography structures, where terminal ends are artificially locked into place by crystal packing lattice forces. As a result, figure 2 serves the visual example of training bias, proving that the AI incorrectly imitates these static crystal artifacts rather than calculating the true flexible physical reality of the terminal sequences freely floating in solution. So now let's validate these models statistically. Figure 3 serves as sort of a reality check. And we're evaluating the exact orientational errors of AlphaFold's nucleic acid predictions by benchmarking them against the giant pool of actual solution state experimental data. First, we have to know what they used as their gold standard metric. And that's an NMR tool called residual dipolar couplings, or RDCs. And so let's think of an RDC as a tiny, highly sensitive magnetic compass needle that's attached to every single chemical bond vector in an RNA or DNA molecule, like the CH or the NH bonds in a nucleotide base. If an AI predicts that a nucleotide base is tilted at a specific angle, researchers can then mathematically calculate what its RDC compass reading should be. They then compare that calculation to the actual RDC measured from a real sample in an NMR spectrometer. And so if the predicted structure is physically accurate, the calculated data and the experimental data should track together perfectly, like a straight line, right? If we graph that out, it'll be a straight line. The R squared would be 1, indicating a perfect correlation. So we see the results of running this RDC check across 28 different DNA and RNA structures in figure 3. And for canonically paired base double helices, standard A-form DNA, RNA, or B-form DNA, the points cluster beautifully along the diagonal line. The AI knows how to draw a standard staircase. The second you look at internal loops and bulges and junction regions, the points scatter wildly off the line. They're way off course. And so this visual fuzziness or dispersion is graphic proof that the AI's math is misjudging the real angles of those nucleotides. This is quantified by cross-referencing with table 2, and that quantifies the root mean square, or RMS, orientational error, which measures the angular mistake in degrees. And here we can see that the average structural error ranges from 7 to 30 degrees. Now, 7-degree error kind of happens in simple, rigid helical stems, but a 30-degree angle, that happens in highly functional loop regions. In structural biology, a 30-degree mismatch is massive, and it completely alters the protein or even a drug molecule, how a drug molecule might dock, right, into the RNA pocket. And that renders the AI model unreliable for active drug design without physical corrections. So if figure 1 showed us a single case study of AI failing to understand its environment, figure 3 shows that this failure is a systematic trend. Now you can see the statistical proof in the paper's subtitle, The Imitation Game. This figure elegantly shows that the AI is highly proficient at imitating standard rigid structures that it's seen thousands of times in its training data. And there's tight correlations, but it systematically miscalculates all the unique, flexible, and non-canonical structures that carry out the actual cellular work. That's the wide scatter and high angular errors. So that's significant. Now, if we look at figure 4, this actually shows us some changes in the Q factor. And this is an important variable in determining the quality of protein structures in structural biochemistry. This is in response to changes that they looked at the Q factor in response to changes in the ionic sodium content. Now, they didn't find any notable increase as the ionic concentration increased. There was no notable increase in the Q factor as the ionic concentration increases. So this also suggests the AI is just not taking ionic interactions into account when determining structure. Lastly, here we have a summary of the overall AI structural prediction of nucleic acids, thus illustrated. And so some of these structural aspects are predicted quite well. Canonical base pairing, common short RNA loops, DNA A tracks. Less common are longer loops, dynamics, details of hydrogen bonding. These are less grounded in reality. There's also an overall consistently incorrect prediction involving the introduction of cations or sequence modifications that might affect structure. And so the validation revealed a broad spectrum of accuracy. While canonically paired double-stranded regions matched solution state really well, the overall root mean square RMS errors in predicted nucleotide bond vector orientations range from about 7 degrees to as high as 30 degrees. And the AI models also failed to capture structural changes dictated by ion coordination or the intrinsic conformational plasticity of loops and junctions. So in conclusion, I have to say that I agree with the authors here. We can say that AI-generated nucleic acid structures cannot yet be blindly trusted for downstream biochemical applications. There remains an absolute necessity for rigorous experimental validation via NMR, RDCs, solution state metrics, or even cryo-EM. And that's to ensure that computational models truly represent the solution state ground truth rather than just an idealized training set replica. Will AI ever replace actual experiment, though, or any computational structural model? Isn't it always better to actually obtain the experimental data instead of just relying on a computational model? I mean, AI can help speed up the process and maybe help us draw some conclusions before we get to the experimental data. But I think experimental data is not going away, and I think we definitely need to look at that very closely and consider that experiment is always better than AI. So I hope you enjoyed this episode of Biochemistry Unfolded, from atoms to algorithms. If you have any comments on this paper, you can find the citation of the paper in the description. If you have any questions, please leave them in the comments below. If you like this video, go ahead and hit the like button and hit the little bell icon so you're notified of updates and that sort of thing. And thank you for watching. I will see you in the next video. You have been listening to Biochemistry Unfolded, from atoms to algorithms. Thank you for watching. We will see you again in the next video.